genomic analysis of a strand of Beethoven's hair, a strand that's unlocking new secrets about his life and death.
For 2 centuries, the world believed it knew Ludwig van Beethoven through the echoes of his music, yet a handful of preserved hair strands carried a different story waiting in silence. When scientists finally decoded his DNA, they uncovered truths that reshaped his identity and exposed how little genetics alone can explain genius. The man behind the myth. Long before laboratories and genome sequencing entered the story, Beethoven existed as a paradox, carved into cultural memory, a composer whose music radiated power even as his own body betrayed him. Born in Bonn in 1770 and later transformed into a symbol of Vienna's musical revolution, he became the embodiment of artistic triumph against personal suffering, especially after progressive hearing loss threatened the very foundation of his career. Letters written during his lifetime reveal a man wrestling with isolation, financial pressure, and fragile health, yet pushing relentlessly toward compositions that redefined emotion and sound. Historians built an image of Beethoven from diaries, accounts of friends, and the mythology that naturally grows around genius, gradually forming a narrative that felt complete, almost sealed by time. Still, beneath the familiar story lingered questions that traditional history could not answer with certainty, including mysteries about his illnesses, his family background, and the private struggles hidden behind public brilliance. Museums preserved locks of his hair as treasured relics, while scholars debated what these fragments might reveal about the man himself, imagining that science could one day confirm what speculation never could. The idea that modern technology might revisit Beethoven's life did not begin as an attempt to challenge his legacy, but as a quiet effort to understand him more fully through evidence rather than legend. That ambition would eventually lead researchers away from concert halls and archives, and toward a laboratory where history and biology would collide in unexpected ways. A lock of hair, a century of assumptions. For generations, historians believed they possessed intimate physical links to Ludwig van Beethoven through several treasured locks of hair, each preserved as if it held the power to illuminate his final years. These relics circulated through museums and private collections, inspiring medical theories about everything from lead poisoning to chronic infections, because researchers assumed the strands offered an unbroken biological bridge back to the composer himself. Throughout the 19th and 20th centuries, scientists repeatedly tested these samples, confident that they were examining authentic pieces of Beethoven's body, and their findings shaped popular narratives about his suffering and decline. Yet many of these claims rested on a fragile foundation, since authentication methods were limited, and provenance chains sometimes relied more on trust than verifiable documentation. As scientific techniques evolved, skepticism grew around whether all these locks actually belonged to Beethoven, especially after inconsistencies emerged between reported results and historical accounts. Some samples displayed chemical signatures that seemed implausible for a man of his time and lifestyle, while others carried trace elements that conflicted with documented details of his medical history. The uncertainty placed enormous weight on the question of authenticity because incorrect samples risked distorting everything scholars believed about his final decade. This tension eventually pushed researchers to re-examine the entire collection, leading to a meticulous investigation into origins, ownership transfers, and microscopic characteristics that could reveal whether a strand truly came from Beethoven's head. When a set of 5 independently sourced locks was finally confirmed as genuine through genetic consistency, the scientific community gained a rare opportunity to study Beethoven through the lens of modern genomics rather than speculation. What began as an effort to clarify disputed relics soon evolved into a turning point, because the authentic DNA offered answers no biography could provide and opened the door to revelations that unsettled long-standing assumptions about his ancestry and personal history. Reading the genome. When researchers finally secured 5 authenticated hair samples with matching genetic markers, they initiated one of the most ambitious historical sequencing projects ever attempted. An international team led by the Max Planck Institute, the University of Cambridge, and several genetic laboratories collaborated to extract and reconstruct Beethoven's genome using protocols designed for highly degraded DNA, normally recovered from archaeological remains. Each strand was treated as both a biological artifact and a historical document because the team understood that any contamination, sequencing error, or misalignment could distort conclusions that would immediately ripple through music history, medical scholarship, and public understanding of the composer's life. Their goal reached beyond simple curiosity, since the genome promised insight into Beethoven's ancestry, inherited illnesses, alcohol-related liver disease risk, and the mystery surrounding his long-debated hearing loss. As the data took shape, the researchers discovered patterns that aligned with some historical accounts, including elevated genetic predispositions to liver complications and certain gastrointestinal conditions noted in his letters and physician reports. Other findings challenged expectations, since the genome offered no clear explanation for his deafness, suggesting that environmental or infectious factors, rather than straightforward hereditary causes, may have played the dominant role in his auditory decline. Yet, the most dramatic moment arrived when the Y chromosome lineage extracted from Beethoven's DNA did not match the paternal line profile of living descendants from his documented family tree. This discrepancy pointed toward an unrecorded paternity event somewhere in the generations preceding Beethoven's birth, a revelation that unsettled genealogists and reshaped understanding of his household's private history. The completion of the genome gave historians a rare scientific vantage point, allowing them to revisit long-standing debates through measurable evidence rather than interpretation alone. However, the results also introduced a new tension by revealing limits in what genetics could explain, pushing the narrative toward deeper questions about inheritance, identity, and the origins of human creativity. Genetics versus genius. As researchers examined the completed genome, they realized that the most intriguing insights were not confined to ancestry or medical vulnerability, but emerged from a deeper analysis of Beethoven's genetic architecture. Using modern polygenic scoring, a method that aggregates thousands of tiny genetic variants associated with specific traits, scientists calculated his predicted capacity for beat synchronization, a measurable proxy for musical aptitude used in contemporary behavioral genomics. The results surprised nearly everyone involved because the score fell comfortably within the ordinary range of present-day populations, offering no exceptional signal that might hint at innate, genetically driven musical brilliance. This single finding challenged a quiet but powerful assumption that genius, especially musical genius, could reveal itself through biological markers identifiable in a genome. Polygenic scores, though statistically meaningful across large groups, cannot predict the abilities of any single individual with precision, and the Beethoven analysis underscored that limitation with remarkable clarity. The genome displayed no distinctive configuration of musicality-linked variants, and no unusual clustering of traits associated with creativity, auditory acuity, memory, or cognitive processing speed. Instead, the data portrayed a man whose genetic predispositions appeared wholly unremarkable when viewed through the lens of modern population genetics, even while his compositions continued to define the emotional vocabulary of classical music. This disconnect between genetic ordinariness and artistic magnitude introduced a philosophical tension, prompting researchers to reconsider how much of human creativity can truly be traced to biology, and how much arises from lived experience, resilience, training, and the complicated interior world of the individual. The revelation also reframed earlier discoveries from the sequencing project, since the identification of a disrupted paternal lineage and the clarification of his medical risks revealed important truths about his life, but offered no pathway toward understanding how he produced work that still resonates across continents and centuries. The science, rigorous as it was, illuminated the boundaries of genetic explanation and set the stage for a broader reflection on the interplay between inheritance and the human environment in shaping extraordinary achievement. What the DNA couldn't explain. The contrast between Beethoven's ordinary genetic predispositions and his extraordinary artistic legacy pushed historians and scientists to revisit the forces that shaped his life beyond biology. His childhood unfolded under the intense pressure of a demanding father who envisioned transforming him into a prodigy, and this early regimen of relentless training forged technical foundations that genetics alone could never predict. Throughout his adulthood, Beethoven navigated emotional turbulence, social isolation, and chronic illness, conditions that often deepened rather than diminished his creative drive, allowing personal struggle to evolve into a distinct musical language shaped by introspection rather than convention. Environmental forces, mentorships, chance opportunities, and the cultural atmosphere of Vienna all contributed to a trajectory that no genome sequence could chart with precision. The medical findings from the genomic study clarified certain aspects of Beethoven's suffering, while leaving others unresolved, since his heightened risk for liver disease aligned with documented alcohol use and autopsy descriptions. Yet the absence of a definitive genetic explanation for his hearing loss reinforced the likelihood of infectious or autoimmune causes. These insights expanded knowledge of his physical decline without offering a singular answer, emphasizing how multiple interacting factors shaped his final years. The investigation also reshaped the narrative of his family history because the unexpected paternal line rupture introduced new complexity into genealogical accounts that once appeared settled, inviting careful reconsideration of relationships and identities within his extended household. Although the genomic project provided unprecedented detail about Beethoven's ancestry and health, it ultimately highlighted that creativity cannot be decoded through biological analysis, since the genome revealed vulnerabilities, lineage surprises, and physiological tendencies, but left the origins of his musical vision untouched. This tension between what science illuminated and what it could not explain reinforced the enduring mystery war at the heart of Beethoven's story, demonstrating how factual clarity can coexist with unanswered questions about human expression, resilience, and imagination. Legacy, rewritten by science. The sequencing of Beethoven's genome transformed him from a distant historical figure into a subject of modern scientific inquiry, revealing how technology can illuminate ancestry and illness with a clarity previous generations could not imagine. Yet these discoveries also demonstrated the limits of genetic interpretation, since the data mapped biological risks and familial ruptures, while leaving the origins of his creative language untouched. The juxtaposition of measurable inheritance and immeasurable artistry invites a broader reflection on how history is written, because objective evidence can refine a narrative without ever capturing the interior experiences that shape a life. Beethoven's story shows that identity emerges from more than lineage or physiology, instead forming at the intersection of environment, perseverance, and personal desire. As genomics reshapes biographical understanding across disciplines, his case illustrates that scientific precision can coexist with mysteries that resist quantification, ensuring that certain dimensions of human expression remain anchored in emotion rather than data. If you want more stories where science rewrites history and challenges what we thought we knew, subscribe and stay with this channel for the next investigation. Share this episode with someone who loves music, and let us know which mystery you want uncovered next.